to make it more practical, I prepared a couple of slides, no more than a couple of slides, in which I tried to outline which are, from a clinical perspective, what may be the inclusion criteria, what may be the outcome, and what can be some statistical thing that we could do. Can I share them so that you can give me your, either I don't know answer, which is perfectly reasonable, or it doesn't make sense answer, which is fine as well. So what we have thought together with Rashmi and Alexei, if I can share a couple of slides, and these are based, you can see them. Can you see them?
Mm-hmm. Yeah.
Can I just try not to put them in presentation mode? Last time it took forever to do that.
Yeah.
So I just stay with this. So there are two phenotypes in this refractory difficult to treat SJIA, assuming that 80% of the patients do respond very well to available biologics, which are IL-1 and IL-6 inhibitor that became available essentially in 2010. So I'll change that because I want to make sure that we review cases in the biologic era where IL-1 and IL-6 inhibitor were available. And one phenotype is the patient with refractory arthritis and persistent chronic arthritis, and I will kind of skip those for the sake of time. Let's stick to the other phenotype, which is much more relevant and new, okay? And these are patients with largely systemic disease, little if any arthritis. They are more or less the same, even if they have little MAS or relapsing chronic MAS or chronic lung disease. They have a biomarker that identifies this population. We have been discussing this in extent, and I think this should be the core of the registry for a natural history study. So, and we think they are phenotypically slightly different, but the same disease with the same pathogenic pathway behind them. And there is plenty of evidence suggesting this. And Alexei, you just jump in anytime you want, please. So I guess the important thing here is era, time. And then we have to enroll them, making a little wide in the registry. We want to have them with or without ILD. Can't really measure interstitial lung disease as we do now, because 10 years ago we had, little idea of what is there, but we can do it. We can certainly try to build a registry and then use a propensity score on the features of the patient enrolled to select those that are comparable to those included in the clinical trial in 2023. Would that be acceptable? Does this make any sense to use a propensity score in this approach when the feature of the patients in the registry strategy may be a little wider? There is no genetic diagnosis as for spinal atrophy, obviously. And then select those that are similar. This is a question for you. 3.
So when you say you, I'm not sure who you is in this case, but
the three experts.
So I am, not a statistician myself, and obviously propensity scores are an important parameter here. There's so many other things as well in terms of the timing of where in the disease process you set your baseline for follow-up, et cetera. So I can't really comment on the exact way that you would do the matching. This is something where we could go through the whole Bettina Hansen presentation where she walked through all of that very carefully. And she, by the way, would be an excellent advisor to you in this setting. But I think basically setting up this kind of a registry with all of these this information in there is a very good start. And you have, it's good that you have the mass criteria already established. And you have some biomarker data, maybe not for everyone, but some biomarker data that I think will be very good. But then, how exactly you extract your external control cohort from this registry, then I can't tell you right now how exactly you would do that. But there are mechanisms to that.
But the approach of making it wide and then select
right,
in a proper statistical manner, is that doable? Because this is not a monogenic disease, so we can't base our selection on the mutations, obviously.
Right. Yeah. And Alagille syndrome, which is the example I used, is also that way. It doesn't have, that definition. So that genetic definition.
And I guess that disease duration is an issue. That's why it's number one in the list here in the slide, because your baseline, it's what defines the timing. And I see the point here.
Yeah.
Alexei, any comment here on disease duration? This is complicated for us.
Right. We did not think about that, but certainly we have to.
Yeah. And to follow up on Yuki's comments, Yuki, I think the CARA registry may have a longitudinal follow-up on some of these patients at least. Yes. Well, that could be a source.
I mean, we have, I'll show you some preliminary data on, the general cohort of patients that we have and the kind of data that we are collecting. But it's very preliminary analysis that we did kind of quickly. But I am confident that we should be able to identify patients with specific phenotypes and potentially enrich for specific phenotypes. And there is also a study that Grant and I are heading up, enrolling lung disease patients in the registry as well. So I think that there's definitely a role that could be played to find, the right kinds of controls for a study like this.
Excellent. I do have another question, and that regards concomitant treatments. So these patients are usually, and we have discussed this already this morning a little bit, but to you, the experts, these patients are on a load of immunosuppressive treatments that go from high-dose glucocorticoids to anything you can think of. Most of them is off-labels, and this will be collected in the registry. So this is an important, can't be the primary outcome, but I understand it's a very important, measure of the efficacy of a novel treatment. You get rid of the drugs. What is the take of both of you that are in the rare disease fields and have seen this, on how to measure this? I mean, this is easily extractable from the chart retrospectively, and it's unbiased, certainly unbiased. Can we use it? How shall we use it? Maybe Kerry?
Yeah, I mean, I can start off with a few thoughts. I think it's critically important to capture concurrent therapies, right? Mm-hmm. It's going to help you both with any conclusions you try to draw, certainly safety effects, as well as efficacy. So I think that it is critically important to capture that. I think the other thing that I would offer, as you are capturing them, this is from my days as a pediatric gastroenterologist, hepatologist, I mean, we weren't always standardized in the manner in which, medications were given. There was a top-up group and a top-down group and a bottom-up group. And different people did different dosings of biologics depending on response, and it wasn't always necessarily, there wasn't an algorithm, a standardized algorithm to follow. And so I think that it's also critically important to sort of capture those types of practices, if that's possible, because that might differ by region, it might differ by health system. So it's just another confounder.
Yeah, Fabrizio, I think to your question, I mean, to your point, and also to Kerry Jo's point, if there is a harmonized or agreed upon or uniformly used background treatments, the standard of care, that would make it a lot easier to make these assessments. And I said specifically to the corticosteroids, use of corticosteroids is an art. It's not really science, and everybody uses them differently. Not only, doses, but also how they are administered, how they are tapered. So this makes it extremely challenging to use changes in background medications to assess efficacy. Again, if we talk about complete discontinuation of steroids altogether and patients are doing great, that's an easier call to make, I would say. I mean, that's kind of a hard or easier situation to interpret. But decrease, say, 50% of steroids or some other treatments, I think it's very challenging. And again, think about this as a proxy to the actual clinical benefit, which is, patients are doing fine clinically. That's really the endpoint. Or they're not, I mean, if we use mortality, which was one of the examples, which are very hard endpoints, that would be, a definitive clinical benefit, and whether whether that can allow decrease of the toxicity of the other medications, that's kind of a secondary benefit.
Would it be helpful to have the same centers that are involved in the trial, let's say, to use the same centers to collect the retrospective data? Because as you mentioned, the use of steroids is the art, and it's certainly different in different centers. Would it make a difference or not? What do you think?
It might, but I think the total dose of glucocorticoids in the area under the curve of the dose of glucocorticoid is a very hard outcome. Again, a 30% or 50% decrease would not, I think it would not be extremely convincing, but Veronica was pretty clear in the table you showed, dramatic efficacy. Isn't that what it's the first line of your table? Yes.
If you have a good treatment effect, it's so much easier than if you have to say, well, they say they're feeling better, but you don't have a way of actually, capturing that. That's so tough. And I grew up in this field, in the HIV field, where we had once we got the viral load, it just became so much easier because it was something you could measure, you could suppress it, you could look at drug resistance. But it's very different with these other chronic diseases where it's so difficult to get that kind of standardized response. But I think that in terms of what I think your question was, like with the different standards of care and different centers, there are ways of then stratifying the analysis to kind of look center by center and accounting for that heterogeneity and standard of care.
I don't think we have enough patients to do that. Sorry, this is practically not doable, and I get the point on this, the different standard of cares of the different immunosuppressants / biologics based on availability in different regions and continents. That was an important thing that we should be aware of and take care of. Kerry, can I ask you your point of view on the total dose of glucocorticoids in children, because we are talking children particularly. Is that something, how important that is? And maybe also Nikolay, what is your take on something that is clinically relevant? 30 to 50% probably decrease is not clinically relevant. Getting rid of glucocorticoids is clinically relevant, Nikolay, to your point. I think I agree. Anything in between, because sometimes, you know, you need those very low daily doses that we rheumatology are in love with. They maintain and they help maintaining their patients, and they are irrelevant from a clinical point of view.
So again, I'll get back to this sort of concept of this being a proxy of the clinical benefit, where patients are doing well for some reason, and that's why you are going down on some of these toxic medications. And I mean, just for more of a conceptual comment, we're talking about lowering the use of steroids because we're lowering the toxicity, sort of the side effects of the steroids. For new drugs, we don't even know what these toxicities could be. I mean, talking about brand new products that are, you know, studied in a handful of patients. So there are uncertainties around the safety of these products too, to say that, you know, we should be replacing the toxicities of steroids with toxicities of some of these newer products. Again, this is just a consideration, but to the point, I completely agree. I mean, I'm a rheumatologist, and we're the last people in the world who want to prescribe steroids. But yes, they work well, and that's why we use them. Now, how we use them, again, I don't think there's a uniform definition or doses, but I think trying to decrease the toxicities of these steroids is a clinically important benefit to patients. But again, it's driven by first a benefit on some clinical outcome, you know, with new intervention. I mean, thinking about TNF inhibitors for, you know, RA, they are steroid sparing, right? Because, you know, people rarely use steroids nowadays because you have these highly effective treatments. But the effects of the treatments is really on suppressing the inflammation or sort of managing the underlying disease. And then the secondary benefit is potentially decreasing the toxicity of some of these treatments.
Got it. Rashmi.
I was just going to point out quickly that actually what we see in the patient group is that after disease control, some patients are able to go off steroids for the sake of managing inflammation, but then they get transferred over to the endocrinologists because they have been on steroids for so many years that they have adrenal issues, and then they often go on hydrocortisone to try to then slowly taper, because they literally cannot taper even if they don't need. So we might need to special case that, because from inflammation perspective, they might be on really low dose, but they can't get off it till the time that their adrenal glands wake up. So that was one slight comment to that. But I think I was going to suggest that for Alexei, maybe present the one slide he has on the retrospective study plan, chart review plan for MAS, so that we just make sure to discuss that. I don't think he's here right now. I think I have that. Fabrizio, do you have that?
Yeah, I have that, but the problem is where?
Okay, hold on. I have that too, actually. I have that too.
Find it.
Yeah, I think I can put it on my screen.
Alexei's there.
Alexei's here. Alexei, do you? I can actually share my screen because I have that up right now. Do we want to quickly discuss that? Yeah, this is the plan for MAS.
Alexei, you want to discuss these slides briefly in the context
of
what we have just discussed?
Yes, we discussed it earlier, but the point is that here we are looking at the MAS component of the disease, which is an acute event. In the existing clinical trials, the main sort of inclusion criteria is the patient should meet the diagnostic classification criteria for MAS, and they are supposed to fail to respond to high-dose steroids. So sort of those are two main points in the inclusion criteria. So the idea is to generate some kind of a historic control group that would, again, satisfy the same criteria. and then compare the time to a complete response. And again, there are several clinical and laboratory features so that there is an overall response. And then we can get a little bit more granular and look at the time to normalization for each of those components in the complete response. Certainly death rate, we know that historically the death rate is somewhere between 10 and 20%. So that certainly would be something that would be easy to compare. But then the next point would be this cumulative exposure to the typical drugs that we treat macrophage activation syndrome. That includes steroids. Again, we will look at the ability to taper, but also we will quantitate the total exposure. In many of those patients, if they fail steroids, we start medications like etoposide, and as you know, it puts you at risk to develop myeloid malignancies in the future. So again, we would look at the proportion of patients started on that type of treatment, and as well as total number of infusions. And the same thing would be done for other typical medications that we would use to treat MAS. And again, so that we would have a historic cohort, right, and our clinical trial. And we specifically would compare these parameters. Do you think it's reasonable?
The definition of complete response for MAS was defined in 4 trials, okay, that are ongoing, but is objectively measurable based on laboratory parameters. There is, however, one part of it which is based on not in this case, but there are response measures that are based on a visual analog scales or on physician global assessment. Can we go back and, define a physician global assessment of disease activity based on chart review? Is this doable? And would this be considered comparable to the outcome measure in a prospective clinical trials? This is a methodological question. And this is true for any measure in pediatric rheumatology that we may apply to this refractory SJIA/MAS group of patients. We're using physician global and parents global. Parents global, we cannot use retrospectively. Physician globals, it makes sense to me that I can judge the severity of a disease retrospectively based on chart review. Would this be methodologically sound?
Kerry, Joe, and I'm just going to sort of start this one off here. And I can't comment really on the specifics related to, SJIA because, or MAS, because this is not my area. But I can tell you your general question of can we go back and use a physician global assessment, which I'm assuming, I don't know the components of your physician global assessment for MAS, but has some subjective elements within it, correct? I mean, it's not just a compilation.
It's based on the evaluation. It's based on the evaluation of a list of clinical symptoms and signs. Mm-hmm. Mm-hmm. But there is
some of which may have been subjective interpretation, right? That becomes really challenging. As you saw the examples that Dr. Miller was giving of where external controls have worked. And resoundingly, what we see is objective endpoints that it is very clear you would not question, hospitalization or mortality, things like that. It becomes really, really hard once you introduce instruments that may have bias, and then add on top of that retrospective review, which can be very difficult to validate. So I'll just say that there are a lot of challenges, and that would have to be really discussed thoroughly with the FDA prospectively.
I'm taking notes.
Please do, because I think these are kind of the questions that we will raise once the question comes to us. I think the examples that used external control are, as Kerry Jo mentioned, based on really objective and hard clinical outcomes, not just lab parameters, for example, like hospitalizations, mortality rates, or the need for ICU care. I think these would be fairly hard and fairly objective endpoints. I think that would make the reliability of the outcome assessments much more robust.
Mm-hmm.
I think,
but the MAS response is based on normalization of lab parameters that are typical of MAS, including ferritin, platelets, white blood cells. So these are objective measures, and are the features, the cardinal features of hyperinflammation. I would consider them very objective.
There's objective measures, and then to underscore Nikolay's point, what you're eventually looking for is that tie, that direct tie to the clinically meaningful outcome, right?
Say it again. Can you speak a little slowly? Because my line is
sure.
Sorry. I'm sorry.
So there's
my fault.
There's objective measurements, but then there's also that direct tie to clinically meaningful outcome, right, or a clinically meaningful benefit. So both of those pieces need to be there.
So, but the days in the ICU would be a meaningful measure of that, correct? It would be something that is clinically meaningful.
Yeah, so I think that's exactly the point. So you're measuring all of these parameters to make a determination whether the patient is getting better, that the patient doesn't require, that level of care. Certainly shortening duration of ICU stay, shortening duration of hospitalization. I think
so.
Would be
things like the need for ventilation, oxygen supplementation, inotropic support for the heart, right? That would be taken seriously, those measures as well, right?
I mean, these are definitely relevant and important outcomes, and I think the case of MAS as sort of a example where you can use external control is probably easier to make, because you have these more acute setting, more urgent emergent situations where you can capture improvement in all of these in a relatively reasonable time frame for a clinical trial. The challenge might come for some more subjective measures for a longer, for refractory, let's say refractory SJIA versus MAS. And again, I'm kind of viewing these as two separate entities and two separate concepts to think about how to extract or use the retrospective data.
I think this is very helpful. I think those are hard outcomes that we can certainly retrospectively derive from patient charts review, and they have to be together with the laboratory evaluation of MAS complete response, Alexei.
Yeah.
This is what is being used as a primary outcome.
And certainly this kind of data could be easily extracted from the medical records, right? So,
yes.
Right.
Presumably, it's Kerry Jo. I just want to jump in here. I don't know if you've all had a chance to see the FDA guidances that have been coming out on real-world data, real-world evidence. A lot of them came out just at the end of this past year. And one of those guidances is one on how assessing electronic health records and medical claims data to support regulatory decision-making. So there's a lot of, I think, good considerations for going about that in that guidance.
But also in this case, you would have the clinicians, the clinical caregivers notes and charts. But then when they go to ICU, that would be a different set of information, right? But do they get merged back together?
You mean
in the clinical records? Yes, yes.
Yeah, okay.
I can accept, if I want you to look at the clinical course while the patient was admitted, I can see both.
Okay. Yeah, and I had all of those guidances listed on one of my slides. That was the website was set updated as of January, 2022. So, but that's why I was also really emphasizing that this is there's a lot going on in this area right now. So I think the more we all learn together, the more updates there will be. But a lot going on, both from the US and European perspective in this area. I was wanted to go back to you, Fabrizio. Somewhere along the line earlier, you mentioned about a difference in clinical presentation in the US versus Europe. I don't know if I caught that correctly.
Not exactly the presentation, is that and Alexei can jump in as much as he wants, obviously, is that there appears to be more patients with interstitial lung disease or lung disease, let's call it, in the US, and they appeared a little earlier. We have just finished to run a survey among European pediatric rheumatology centers, and there are coming up, I shouldn't say plenty of, which is not true, but dozens of patients are showing up also in Europe. And we are seeing a couple of them recently. So they are also here. It looks like the similar story with secondary amyloidosis in the past. You may not have heard of it, but secondary amyloidosis was secondary to chronic inflammation. It's very frequent in Northern Europe. And the genetic background of people from Northern Europe are pretty similar to the white Caucasian in the US, right? But there was essentially no secondary amyloidosis in the US. God knows why. So there are environmental differences. I don't think it's genetics. There are environmental differences. But these patients that we thought we didn't have until a few years ago are actually here as well.
Okay.
And I don't think we were missing them. I think that they weren't here.
No, I think there are geographic differences in the prevalence of this lung disease that do not necessarily correlate, by the way, with the use of biologics. So it's perplexing, yeah.
This is also, I think, a critical and challenging component of utilizing these external data sources is ensuring that you actually have a collection of patients that is representative of the totality of the experience of the disease. It's really hard to do, given all the differing practices.
But it's based on the inclusion criteria of the natural history study, which may be wider than what you actually are going to enroll in the clinical studies. And then you
absolutely. No, absolutely.
Okay.
But I'm just responding to the fact that it sounds as though there's patients showing up that, I mean, they may be there, they're just not being registered in the same way in Europe as they are here. So it's just.
There are expert pediatric rheumatologists in Europe. We should not have missed them. The past, the clinical phenotype is so severe that it's very improbable that we missed them. And Yuki was the ones who, picked up this phenotype in the first publication in 2013. And it wasn't there before, I think. It's difficult to say.
Well, there were, individual cases reported over the years, but nothing in terms of the numbers that we were seeing since 2005, which is what was a concern.
If I may ask a question, are there examples of trials, etc., where parents, patient groups collected data? I mean, there are, the NORD has a registry, patient-driven registry, and we've been looking and talking to NORD actually about doing an SJIA one. But the question really is that, we have limited bandwidth and energy, and which one do we want to what do we want to do? Like, would that help in this kind of endeavor, or is that not the sort of data that might help?
Are you, I'm just going to put this out there. Are you guys familiar with the Rare Disease Cures Accelerator Data and Analytics Project?
Not that one. No, I don't know.
What so it's an FDA-funded effort in conjunction with the Critical Path Institute is doing it, and NORD. And essentially, we have built a platform that's like an integrated database and analytics hub that can collect information from all types of sources, registries, patient records, trials, and it takes them all in, standardizes them, and essentially then is available to use for people to use and inform, like to get aggregate data about a condition down to the patient level to inform trial design, et cetera. So that is from my understanding, it doesn't take a huge lift on those who are providing that data. So I would encourage you guys, as you think about trying to get the most data possible together in a repository, so that it can be sort of used to make some of these determinations and inform the natural history and inform trial design, that you take a look at that effort. And I'll see if I can find a link to it up in the chat.
Yeah, because we just spoke to NORD about a month ago, and we got, we found out whatever they could refer us to. I'll make a note of this.
Yeah, you may have spoken to NORD, but for this, it's actually being housed in the Critical Path Institute. So I would contact them.
Right. Yeah, I was just going to mention that as well, Kerry, Joe. So absolutely. And it's built in such a way that, exactly as you explained, that it can serve, all the different diseases. And very collaborative. It's quite an amazing project.
Yeah. So that was one question. The second was, I wanted to, after this, we have a session which we couldn't fit in before, where Grant is actually going to help, like kind of with Yuki and Bas, etc., to try to figure out previous studies and if we can extract data from that. And Veronica, are there examples you found of such data being used as external control derived from previous research, etc.?
So I think the ones out of those examples in this paper, they were all mostly for the first indication or sort of the first drug for a disease. So I don't know to what extent other trials were available for these, for the ones that were approved using external controls. I know certainly in other diseases, we're discussing that a lot. But I think for rare diseases, so many of these are the first drugs for the disease. So there aren't really any other treatment trials available. But that's why I think it's so important to transition from natural history studies to on treatment prospective natural history, prospective observational cohorts, so that information can be collected. But I think for most, there's some rare diseases, obviously, where we have more than, more than one treatment. But, my understanding is the vast majority, this will be the first time. Does that make sense, Kerry?
Are there other questions people have? Want to give a chance to the community?
A lot of thoughts and a lot of reading to do here. Thanks. Thanks, Kerry, for sharing the two sites, the two links. That's very important for us.
No problem. I'm really happy to support this effort. It can make such a tremendous impact on drug development to really see the community getting together and working with the FDA and all other stakeholders. It takes all of us.
It does. Actually, I don't think you were here in the morning. So let me just, from the patient perspective, here's the dilemma we find ourselves in. We're kind of the ultra rare phenotype within a rare disease. So the rare disease has 2 drugs approved. So, and we are kind of lost in the error bars till the time the foundation started focusing on it and started saying, hey, not everybody got better. You know, there's people out there, and we started finding the patterns. So every conference we've had, we've been able to define those conditions more and more and more. But still, I think the dilemma of the trials is that, you know, like you pointed out, it was the first trial for that rare disease. Yeah. In some sense, if there is a trial which is focused on this subset, it'll be the first trial for that subset. But the standards that are for us are actually for a disease which already has 2 drugs.
Mhm.
So that makes sense. So I think we are really like, you know, I listen to all this and I keep on wondering that, is it still a better strategy for us to define ourselves as the ultra rare disease or be a part of the rare disease? Because like there's, you know, some of the asks are so big that I don't know if it will help the current generation of children, and we're just going to have to wait. And it's not because the need isn't there. It's not because there aren't, that we haven't understood enough in the last 5 years to think of potential drugs that could help us, but there is simply no path.
Mhm.
And so I don't know. I mean, like, you know, we see the ultra rare diseases actually being able to start doing small, very small, you know, starting with N of 1, then going on. But those paths in some sense are not like relevant for us because we are kind of part of a larger rare disease, which already has had trials. So I don't know. I mean, it's a dilemma as to how to approach this and what is the right path. And, you know, how to do these, try to do these natural history studies, whether keep on trying to group with SJIA and then find the subsets of SJIA who don't respond, or to go in for the IL-18, you know, so.
It's all a matter of disease definition, right? How you define this disease and, you know, diagnosis, differential diagnosis to the larger set.
I think you're right, Veronica, but I think we have the instruments to do that now, particularly with the biomarker. So I think we can, Rashmi, and I think it's brand new. Yes, we are inventing something that nobody has done before. That's fine, but I think there is a great group of great group of patients and parents that can drive this forward. This doesn't happen very often. At least I didn't see it happening in my life. So we make it. I'm pretty confident, particularly after today's session. I think we have learned a lot, and we have got a lot of feedback.
Yeah, no, we have learned a lot, I think. And
I think you should look into the last website that Kerry sent. It's for you. It's not now, but you should copy and paste it and look into the.
Oh, we are saving all the Zoom chat, so we'll have that. Lucienne,
but highlight the link, because I did so. Okay. I did
right.
look into it while talking, and I think it's something we should look into, and particularly you should, Rashmi. John should.
Well, I mean, I can say that if there's no other questions, we can wrap the session up and then continue on to the working sessions. Sorry, no, the extra panel on the external data. And I know that, you know, Kerry Jolie at least has to be leaving us, you know. I think she's, so thank you. But thank you so much to all of you for being here with us and helping us think this is literally going to save us like, 6 months of like, trying different things to have this kind of guidance. I think there were a lot of questions answered about. So it's really, really helpful.
Totally agree.
Thank you so much.
Thank you for the opportunity. Good to meet everyone.
Yeah, thank you.
Yeah,
thank you.
and I specifically,
Grant, it's all yours.
I think
I specifically want to thank Kerry Jo for finding the time to meet with us, because you wouldn't have gotten these responses or details from me. She certainly has more relevant experience for this discussion?
Just broader experience. You have the more relevant specific experience.
Thanks.
Thank you.
Thanks.
Thank you.